Welcome to our exploration of the polymerase chain reaction, or PCR. This revolutionary technique has transformed the field of molecular biology by enabling scientists to amplify tiny amounts of DNA into millions of copies, all within a simple test tube. Today, we'll journey through the history, chemistry, mechanism, and applications of PCR, uncovering why it's such a powerful tool in genetics and diagnostics. Let's begin by understanding the origins of this groundbreaking method. The story of PCR begins in the mid-1980s with Kary Mullis, a scientist whose curiosity led to a major breakthrough. In October 1985, Mullis introduced the concept of PCR to the scientific community at a conference. His idea was simple yet profound: create virtually unlimited copies of a specific DNA sequence in a test tube. This innovation opened new doors for genetic research, diagnostics, and forensic science, making DNA analysis faster and more accessible than ever before. At the heart of PCR lies the principle of DNA hybridization. This process relies on the complementary pairing of DNA bases, adenine with thymine, and cytosine with guanine. In molecular biology, this property is essential for copying and amplifying genetic material. PCR leverages this chemistry to ensure that only the target DNA sequence is replicated, making the process both precise and efficient. PCR is essentially a test tube system for DNA replication. It allows scientists to selectively amplify a target DNA sequence, increasing its quantity by several million-fold in just a few hours. This rapid and efficient process has become a cornerstone in laboratories worldwide, enabling everything from genetic research to medical diagnostics. The mechanism of PCR involves a series of enzyme-mediated reactions. These reactions are orchestrated to produce copies of the entire genome or a specific DNA region. The process is highly controlled and relies on the unique properties of enzymes to ensure accuracy and efficiency. Each cycle of PCR doubles the amount of target DNA, leading to exponential amplification. A key player in PCR is the enzyme DNA polymerase. This enzyme is indispensable for the amplification process, as it synthesizes new DNA strands by adding nucleotides to the existing template. By using just one enzyme, PCR simplifies the replication process, making it both robust and reliable for a wide range of applications. PCR requires specific starting points called priming sites. These are short sequences on the DNA where primers, short pieces of DNA, attach. The primers are designed to bracket the target region, ensuring that only the desired DNA segment is amplified. One primer binds to one strand at the beginning of the target, while the other binds to the opposite strand at the end, setting the stage for precise replication. The design of primers is crucial for PCR success. Each primer is complementary to a specific sequence at the edges of the target region. By carefully selecting these sequences, scientists can ensure that only the intended DNA fragment is amplified, avoiding unwanted byproducts and increasing the accuracy of the results. PCR operates through a series of repeated cycles, each consisting of three main steps: denaturation, annealing, and extension. During denaturation, the double-stranded DNA is heated to separate into single strands. In annealing, primers bind to their complementary sequences. Finally, during extension, DNA polymerase synthesizes new DNA strands, doubling the amount of target DNA with each cycle. The first step in each PCR cycle is denaturation. Here, the reaction mixture is heated to a high temperature, typically around 94–98 degrees Celsius. This heat breaks the hydrogen bonds between the DNA strands, resulting in two single strands that serve as templates for the next steps. Denaturation is essential for making the DNA accessible to primers and enzymes. Following denaturation, the reaction is cooled to allow primers to bind, or anneal, to their complementary sequences on the single-stranded DNA. This step typically occurs at temperatures between 50–65 degrees Celsius. The specificity of primer binding ensures that only the target region is amplified in the subsequent extension step. The final step in the PCR cycle is extension. DNA polymerase extends the primers by adding nucleotides, synthesizing new DNA strands that are complementary to the template. This step usually takes place at around 72 degrees Celsius. As a result, the amount of target DNA doubles with each cycle, leading to exponential amplification. PCR relies on a process called thermal cycling, where the reaction mixture is repeatedly heated and cooled to facilitate the denaturation, annealing, and extension steps. Modern thermal cyclers automate this process, making PCR both efficient and highly reproducible. This automation has made PCR a routine tool in laboratories worldwide. One of the most remarkable features of PCR is its ability to exponentially amplify DNA. With each cycle, the number of DNA copies doubles, quickly transforming a tiny amount of genetic material into millions of copies. This exponential growth is what makes PCR so powerful for detecting even the smallest traces of DNA. The specificity of PCR comes from the careful design of primers and the precise conditions of the reaction. By targeting unique DNA sequences, PCR can distinguish between closely related genetic material, making it invaluable for genetic testing, pathogen detection, and forensic analysis. One major advantage of PCR is its ability to detect mutations associated with genetic diseases. Whether it's insertions, deletions, or point mutations, PCR can quickly and reliably identify these changes, providing critical information for diagnosis and research. PCR is incredibly sensitive, capable of detecting even tiny amounts of DNA. This makes it an essential tool for identifying pathogens like the human immunodeficiency virus, HIV, and for analyzing genetic anomalies in small or degraded samples. Its sensitivity has revolutionized diagnostics and research. In medicine, PCR is used to diagnose infectious diseases, monitor genetic disorders, and guide treatment decisions. Its speed and accuracy have made it a standard technique in clinical laboratories, helping doctors provide better care for their patients. Beyond diagnostics, PCR is a fundamental tool in research. Scientists use it to clone genes, study genetic variation, and explore the functions of specific DNA sequences. Its versatility has made it indispensable in fields ranging from genetics to evolutionary biology. PCR has also revolutionized forensic science. By amplifying tiny amounts of DNA found at crime scenes, investigators can identify suspects, exonerate the innocent, and solve cases that were once considered unsolvable. Its precision and reliability have made it a cornerstone of modern forensic analysis. In summary, the polymerase chain reaction has transformed science and medicine. From its invention by Kary Mullis to its widespread use today, PCR remains a powerful, reliable, and versatile technique. Its ability to amplify DNA with speed and precision continues to drive advances in research, diagnostics, and beyond. Thanks for joining us on this journey through the world of PCR.